let's do a quick review of blood supply as it comes into the kidney. So we start from the heart. The aorta descends down through the thoracic cavity in the abdomen, and then we branch off into the renal artery, which branches into segmental arteries, then interlobar arteries, which go between these lobes, and then arcuate arteries, which arc above the renal pyramid, and then cortical radiate arteries, which radiate out into the cortex here. Right? This is another look at cortical radiate arteries. And if you look really closely at these tiny little vessels that come off of the cortical radiate artery, those are called afferent arterioles. And afferent arterioles lead to a glomerulus. Just like you may remember from Bio 151, afferent means incoming in terms of neurons, right? Sensory neurons are also called afferent neurons. Afferent arterioles are incoming toward the glomerulus. There you have filtration, and whatever is small enough to fit through the fenestrated capillaries gets captured by the glomerular capsule and goes through the tubules. What's big enough to stay in the blood leaves through the efferent arterioles, right? Just like efferent neurons are motor. They leave the central nervous system. Those efferent arterioles then lead to capillaries. Now, most of these capillaries are called peritubular capillaries because they go around the tubules and they exchange with the tubules, right? And that's how we modify what is in the filtrate before it goes on to become urine. But some of them are also called vasorecta. Now, vasorecta are connected to the peritubular capillaries, but they go lower. And you find them specifically in juxtamedullary nephrons. And if you remember from last video, juxtamedullary nephrons are next to the medulla, juxtamedullary, and they have a much longer nephron loop, which goes way down, right? And then comes back up. And these are largely for reabsorbing water, these vasorecta. So again, they're part of the peritubular capillary system. It's essentially the same thing, but you find them on the juxtamedullary nephrons, and they are down on this nephron loop. Now, blood leaving the veins is going to have a very similar pathway to blood entering the veins. Those peritubular capillaries or vasorecta are going to dump into cortical radiate veins, which are going to dump into arcuate veins, which are going to dump into interlobar veins. This should sound very familiar compared to the arteries on the way in. But those interlobar veins are going to dump right into the renal vein. There are no segmental veins. So that's one difference between the arteries and the veins. Then the renal vein will dump into the inferior vena cava, which, as you know, dumps right back into the right atrium of the heart. You have over 1 million nephrons in each kidney. So well over 2 million total if you have 2 kidneys. If you don't, your one kidney tends to get larger to compensate for the lack of a second kidney. And all of those nephrons are innervated by neurons. And those neurons are going to be part of the sympathetic and parasympathetic nervous division, right? So the sympathetic nervous division, our fight or flight, causes reduced filtration in the kidneys, right? Who cares how clean our blood is if we might be dead from this, Joe Exotic tiger that's attacking me in the next minute or so. Parasympathetic, our resting and digesting nerve division, increases the blood filtration, and we regulate that based on the amount of blood pressure that we send to the glomerulus at any given time. And we'll talk about that a little bit more momentarily. So parasympathetic nervous division means more urine production, more blood filtration. Sympathetic nervous division means less urine production, less blood filtration. So what does our urine look like? Well, we're getting rid of nitrogenous wastes, and that includes uric acid, which is formed when we break down RNA and have nitrogenous waste. We also have nitrogenous waste from creatinine. If you recall from last semester, creatine phosphate was a molecule that we used to make ATP very quickly in the muscles when we needed it. That's also a nitrogenous product, and when we break it down, we create nitrogenous waste. But the most abundant and most important nitrogenous waste is urea. And urea is created when we break down amino acids. We talked about in chapter 23, when you break down amino acids, you have to deaminate them, meaning take the amino group off, the NH2, and that becomes ammonia, which we then transform into urea, which is a less toxic version. So if we look at the contents of urine versus the contents of blood plasma, you'll notice that your blood plasma has a very tight range of, for instance, sodium or potassium, where your urine has a much larger range. Well, that's one of the ways we maintain this tight range in your blood plasma is by when you have a lot of sodium, for instance, you just ate some McDonald's French fries, you're going to get rid of most of that in your urine, and your urine levels of sodium are going to go way up. When you've been on a low-salt diet for a while, you want to keep as much sodium as possible in your blood plasma, so you're going to have very little in your urine. Same with potassium, same with a lot of other nutrients that go up and down in your diet. Things that are nutrients like glucose, lipids, proteins. Note that we have a fair amount of these in our blood plasma and almost nothing in our urine. That's because this is stuff that we want to keep. Right? And for some of these things, like lipids and proteins, well, they're too big to get filtered out in the glomerulus, and so they just stay in the bloodstream. But glucose is small, and it gets filtered through the glomerulus and into the filtrate, and we have to actively pump it back in order to keep it in our bloodstream. Right? But the urine, usually you're going to have almost no glucose. If you do have glucose in your urine, that's a sign that either you just ate a whole lot of cotton candy, or you might have something else wrong, like diabetes, where you have just so much sugar in your blood that the pumps that pump it back towards your blood from your filtrate are overwhelmed. Back here we have nitrogenous waste, right? We want very little of this in our bloodstream, and so we dump a lot of it in the urine. So let's review the functions of a nephron, and then we'll walk through the nephron anatomically and talk about where and how those functions occur. So the first function of the nephron is filtration, and that occurs in the glomerular capsule. Right? Blood enters the glomerulus, those capillaries that make up the glomerulus, and it has a lot of pressure, and that pressure, that hydrostatic pressure, pushes a bunch of stuff out into the filtrate, which is captured by the glomerular capsule, right? It's just like, if you have, I imagine, a balloon that you've put on a hose, right, to fill up a water balloon, but it's got a bunch of holes in it, and so stuff's squirting out everywhere, but you've got this capsule around it to capture all that fluid that comes out. But not just fluid, things that are small enough, solutes that are small enough to move through these fenestrations, these holes. That's filtration. Then we have reabsorption and secretion. Reabsorption is moving stuff from the filtrate back into the bloodstream. Things like glucose, which are small enough that they move through during filtration, but they're valuable, right? That's energy, and so we pump them back using transport proteins into the peritubular fluid. Again, pumping. That means that this is often expensive, right? Sometimes we're moving these down their concentration gradient, but sometimes we're moving them up their concentration gradient. Right? This isn't necessarily cheap, especially on this side for secretion. Secretion is when we take things that did not leave the blood during filtration. They were too big, and we pump them out into the tubular fluid. And these are often things that are like metabolic wastes or poisons, or things that are toxic, or drugs or medications that you may have been taking. Filtration is going to happen exclusively here at the renal corpuscle, right, between the glomerulus and the glomerular capsule. That's the only place that filtration occurs. Reabsorption and secretion are going to happen at various places throughout the tubular system, the proximal convoluted tubules, the nephron loop, and the distal convoluted tubules. You're going to have a lot of reabsorption, particularly at the proximal convoluted tubules and also in the nephron loop. Taking a closer look at the renal corpuscle, you can see the glomerular capillaries here that are surrounded by what are called podocytes. These purple cells are extra cells that sort of support the simple squamous epithelial cells of the capillaries and make them sturdier because there's a lot of blood pressure coming in. And they have, they're called podocytes because they have these little feet that stick out. And there are spaces in between the feet where fluid and solutes can squeeze through. This right here is the afferent arteriole where blood comes in. This is the efferent arteriole where blood comes out. It's a little smaller because you have less fluid than you had coming in, because a lot of that fluid gets pushed through here and gets captured by the glomerular capsule. So this is the capsule, and you have this capsular space in between the glomerulus and the glomerular capsule where fluid is caught, and then it leaves through the proximal convoluted tubule. This is the first part of the proximal convoluted tubule. By the way, note a couple of things. First of all, the distal convoluted tubule also swings back past here, right? And I know it didn't in those figures that we looked at earlier, but those sort of straightened out the glomerulus to make it look like it was sort of unidirectional, but really it swings back around this way. I'll show you an image soon. The distal convoluted tubule comes back around the glomerulus. Also, this patch of cells here that are next to the afferent and the efferent arterioles in the distal convoluted tubule, these are cells that make up the juxtaglomerular complex. Again, that means next to the glomerulus. And that juxtaglomerular complex or juxtaglomerular apparatus detects blood pressure, and when blood pressure drops, it secretes renin. We talked about renin earlier, but we didn't talk about exactly where it was secreted within the kidney. It's secreted right here. Renin, which stimulates a bunch of different mechanisms to raise your blood pressure once your blood pressure has dropped. So if we look more closely at this, so here we've zoomed in, you can see exactly what the filtrate has to pass through to get out of the blood and into the capsular space here. It has to pass through these capillaries, and then you have a basement membrane essentially below those simple squamous epithelial cells, and then you have these podocytes. It says pedocells because those are the feet of the podocytes, right? Come around and support this whole layer. But in between, you have these filtration slits where fluid can get through the holes in those fenestrated capillaries and through the spaces between the pedicels. Now you may remember from when we talked about capillary exchange that there are a couple of forces that are very important. There is hydrostatic pressure, which is just water pressure on the walls that contain the water. And in this case, this is blood pressure, right? You have a lot of hydrostatic pressure in your blood vessels, especially in here because of your heart creating pressure for your blood. And you also have osmotic pressure, right? Water is pulled towards solutes, and you have a bunch of big solutes, especially these colloid proteins in here that can't pass through, and so the water moves toward them. In this case, your glomerular hydrostatic pressure is normally about 50 millimeters of mercury, where your colloid osmotic pressure is about 25. And then we also need to remember that you do have some fluid out here that pushes on these walls, and that is hydrostatic pressure as well, but coming from the capsule, and that's about 15. And so we end up with about 40 millimeters of mercury pushing back and about 50 pushing out. So there's a net of 10 millimeters of mercury of pressure pushing out. You don't need you to remember those numbers, but I do want you to remember those forces. So ultimately, if you drop your blood pressure or your glomerular hydrostatic pressure to where it's no longer higher than these things, well, you're not going to be pushing any fluid out here, and you're not going to be creating any urine, and you're not going to be filtering any blood. And this is one of the big issues that can pop up when you have a big drop in blood pressure. Just like your cardiac output is the amount of blood that pumps out of your heart in a minute, the amount of blood filtered by your kidneys in a minute is called your glomerular filtration rate. And you average about 180 liters a day in glomerular filtration. This is a lot of fluid, a lot of blood that's coming through your kidneys. And it's regulated essentially by the blood pressure that comes through your glomerulus. And you can regulate that a little bit on the local level, but a little bit on the global level, the whole body level, using both nervous and endocrine responses. And we will talk about those in the next video.